Hello everyone and welcome to today's session, and today we'll be discussing the NCERT Exemplar questions for the chapter Human Health and Diseases. This is the disclaimer. So let's have a look at the objectives of today's session. So today we'll be discussing the basic concepts related to the chapter Human Health and Diseases by working through NCERT Exemplar questions. So these questions, they are based on some concepts. We'll discuss this concept by discussing these questions. Okay, so these questions have been divided into these following categories. So we have multiple choice questions, very short answer type question, short answer type question, and long answer type questions. So let's begin with this question. The substance produced by a cell in viral infection that can protect other cells from further infection is. So you have four options. You have to select the correct answer from these options. So here the first option is serotonin, second is colostrum, third is interferon, fourth is histamine. So what will be the correct answer? Option number C is the correct answer here, interferon. Now let's have a look at the explanation. So these interferons, they come under the cytokine barriers. Cytokinins are like messengers that help cells communicate during an immune response. Interferon, they are a special kind of cytokine that play a key role in protecting from the viruses. Now, these are the proteins secreted by virus-infected cells, which protect non-infected cells from further viral infections. Now, let's look at the other options. So, serotonin. This is not involved in fighting viruses. It is actually a neurotransmitter. Okay, low level of serotonin can lead to depression. Okay, but serotonin is usually involved in regulation of mood. Okay, now next one is colostrum. So this is the first milk produced by mother after the childbirth. So this colostrum is very nutritious. It consists of antibodies IgA, which give protection to the newborn against diseases. Next is histamine. Histamine is also a neurotransmitter, but histamine is involved in inflammatory and allergic reactions. Next question, transplantation of tissue or organs to save certain patients often fails due to rejection of such tissues or organs by the patient. Now, which type of immune response is responsible for such rejections? We have the options: autoimmune response, second is humoral immune response, third is physiological immune response, and fourth is cell-mediated immune response. What will be the correct answer? Let's have a look at the answer. Option number D, cell-mediated immune response. This is the correct answer. Okay, so transplantation is the replacement of diseased organ or tissue of an individual with a healthy organ or tissue of same or another individual. Now, the body's immune system acts like a security guard, always on the lookout for anything unfamiliar. If the transplanted tissue is recognized as foreign, the immune system will try to attack and remove it. So this is the main reason why some transplant fail. Now, a specific part of immune system responsible here is the cell-mediated immune response. So this involves T lymphocytes. So cell-mediated immune response is mediated by T lymphocytes, and basically the organ rejection is due to cell-mediated immunity. Okay, so this type of immune response recognizes the body's non-cells or other tissues or organs from other individuals as foreign antigens and cause rejection of the grafts. Next question, antibodies present in colostrum which protect the newborn from certain diseases is of. So options are IgG and type, IgA type, IgD type, and IgE type. So what will be the correct answer? Correct answer is option number B, IgA type. Now, Colostrum, this is a yellowish fluid, okay, produced by the or produced in the first few days by the mother after the birth. Now, this colostrum is very nutrition and consists of antibodies as well. Now, immunoglobulin G, this is actually the most abundant antibody in the blood. Clear? So, IgG immunoglobulin, which is antibody, is most abundant antibody in human and found in serum, whereas IgA is the second abundant antibody, where it is found in saliva and tear also. Now, let's talk about IgD and IgE antibody. So, this constitute of total antibodies which are found in most cells and serum. Now, tobacco consumption is known to stimulate secretion of adrenaline and noradrenaline. The component causing this could be. So options are nicotine, tannic acid, curamine and catechin. What will be the correct answer? So let's have a look at the answer. Option number A, nicotine is the correct answer. Now, tobacco has nicotine, which stimulates the adrenal gland to release adrenaline and noradrenaline, which in turn increases the blood pressure and heart rate. Now, let's talk about tannic acid. Tannic acid is not the answer here. So, tannic acid is actually a type of polyphenol and it's mainly used as a mordant in dyeing. Now, another option, curamine, which comes actually from curcumin, is used as a pain reliever, and catechin from catechu is an antioxidant. So neither of these substances stimulates adrenaline or noradrenaline. Next one, antivenom against snake poison contains. So what are the options? Okay, antigens, antigen, antibody complexes, antibodies and enzymes. So what will be the correct answer? Option number C, antibodies is the correct answer. Now, the snake venom is a biological product. Okay, snake antivenom is a biological product and typically consists of venom neutralizing the antibodies. And this is derived from the host animal such as horse or sheep. It is important to remember that antivenom is not an antigen. Okay, an antigen antibody complex or an enzyme. Only antibodies are used. Okay, so basically these antivenom consists of the antibodies. And these antibodies are used as they specifically target and neutralize the venom. Next question, which of the following is not a lymphoid tissue? So options are spleen, tonsils, pancreas and thymus. What would be the correct answer? Okay, option number C, pancreas is the correct answer. So pancreas is not a lymphoid tissue because pancreas is mainly an endocrine and an exocrine gland, so it is a heterocrine gland. It has two jobs, two functions. It releases hormone directly into the blood and it also produces digestive juices that go into the digestive tract. Now, one of the pancreas' main function is to produce digestive enzymes and hormones like insulin and glucagon. Insulin and glucagon help control our blood sugar levels, while the digestive enzyme help break down the food. Now, the pancreas does not play a direct role in immune response. Unlike lymphoid tissues, it doesn't help fight off germs or produce immune cells. So here you can see the types of lymphoid organs. So you have primary lymphoid organ and secondary lymphoid organ. Primary lymphoid organ consists of bone marrow and thymus, and secondary lymphoid organ includes spleen, lymph node, tonsils, paired passage of small intestine and appendix. Next question, which of the following gland is large size at birth but reduces in size with aging? So what are the options here? Pineal gland, pituitary gland, thymus gland and thyroid gland. What will be the correct answer? Okay, so let's have a look at the answer. Option number C, thymus is the correct answer. Let's have a look at the explanation. So thymus is a lobed organ located near the heart just beneath the breastbone. Now thymus is quite large when we are born, but it gradually shrinks as we get older. By the time we reach puberty, it becomes much smaller. In contrast, other glands like the pineal gland, pituitary gland and thyroid stay about the same size throughout life. The pineal and the pituitary are both in the brain and the thyroid is in the front of the neck. So here you can see in this image, you can see the location of thymus. Next question, hemozoin is. So you have the options: precursor of hemoglobin, toxin from Streptococcus, toxin from Plasmodium species, or toxin from the Haemophilus species. So what will be the correct answer? Okay, option number C, toxin from Plasmodium species. This is the correct answer. Okay, basically hemozoin is a toxin which is produced when the Plasmodium digests the hemoglobin present in the RBCs. Then the hemozoin is produced. Okay, when the RBC they burst, then the hemozoin is released, and this hemozoin is responsible for the chill and high fever recurring every 3 to 4 days. Okay, this is clear. So when is hemozoin produced? It is produced by the digestion. This digestion is done by the Plasmodium. Okay, digestion of what? Digestion of the hemoglobin present in the RBCs. Now, to continue the life cycle, Plasmodium enters the human body as sporozoites, which is the infectious form of this pathogen, and they multiply within the liver cells. Okay, and then within the liver cells, these merozoites are produced. These merozoites then infect the RBCs, and then you can see when the RBC is burst, then the hemozoin is released. Next question, one of the following is not the causal organism for ringworm. So what are the options here? Microsporium, trichophyton, epidermophyton and macrosporium. What will be the correct answer? Okay, option number D, macrosporium is the correct answer. Let's have a look at the explanation. Now, ringworm infections are caused by the fungus from the genera Microsporium, Trichophyton and Epidermophyton. Now, Macrosporium is an ectomycorrhizal zoosporic fungus causing disease of economically important vascular plants. Okay, next question is, why is it that during changing weather, one is advised to avoid close, crowded and air-conditioned places like cinema hall, etc.? So we are discussing very short answer type questions now. Okay, so during changing weather, one is advised to avoid crowded places. Okay. Now, because changing seasons are the time when infectious agents are more prevalent, as moist conditions favor pathogens to grow fast. People are more vulnerable during this time as their body system is busy in adapting the changing environmental conditions of temperature, humidity. Now, as a result, they get infected by these pathogens more easily when exposed to crowded places where infectious agents can quickly spread from person to person. Okay, so let's have a look at the next question. Lymph nodes are secondary lymphoid organs. You have to explain the role of lymph nodes in our immune system. So let's have a look at the answer. See, lymphoid organs. Okay, they are part of the immune system. So our immune system is made up of special organs called lymphoid organs. So these organs work together to help our body fight off the germs. Now the organs, okay, where the maturation and proliferation of lymphocytes occurs. These are the lymphoid organs. Lymphoid organs are divided into two main types. Okay, so we have primary lymphoid organ and secondary lymphoid organ. So primary lymphoid organ, it includes bone marrow and thymus, where B and the T lymphocytes mature and acquire their antigen specific receptors. Means now they can recognize a particular antigen. Now secondary lymphoid organ, these organs, okay, are where the lymphocyte interact with the antigen and proliferate to become effector cells. Example of secondary lymphoid organ are spleen, lymph node, tonsils, spare patches of small intestine, and the appendix. Let's continue with discussion. So lymph nodes are small solid structure present at different points along the lymphatic system. Now the lymph node trap microorganisms or other antigens that enter the lymph and tissue fluid. When antigens are trapped in the lymph node, they activate lymphocytes and produce an immune response. Okay, so next question is about the antibody molecule H2L2. Okay, why is an antibody molecule represented as H2L2? Let's have a look at the answer. So each antibody molecule has 4 peptide chains, 2 heavy chains and 2 light chains. Okay, now the 2 small chains known as the light chain, they are represented by L, and the 2 longer chains called the heavy chains, they are represented by H. So an antibody is represented by H2L2, 2 heavy chains and 2 light chains which are linked together by disulfide bonds. Next question, what does the term memory of the immune system means? Let's have a look at the answer. See, when our body meets a particular pathogen like a virus or bacteria for the first time, it responds by making antibodies. Now this response is of low intensity. But if the same pathogen tries to invade again, the body reacts much more quickly and powerfully. Now this is called as a heightened immune response. Okay, this is the secondary response and this is highly intensified. Now this ability to respond more strongly the second time is because our body has a memory of the first encounter. This memory, okay, is due to a special kind of cell called the memory T and the B cells. Okay, so this type of secondary immune response is elicited by the memory T cells in the B cell, which keep ready to mount a rapid and vigorous attack as soon as the same pathogen infects the body again. The next question is, if a patient is advised antiretroviral therapy, which infection is he suffering from? You have to name the causative organism. So let's have a look at the answer. The patient is suffering from AIDS. So AIDS stands for acquired immunodeficiency syndrome. Okay, the causative agent is HIV, which is a retrovirus, means it has RNA as the genetic material. Now next question is short answer type question. So question is, what is secondary metabolism? So let's have a look at the answer. See, secondary metabolism is also called specialized metabolism. This means it is a set of chemical pathways that go beyond the basic needs of the organism. Now, these secondary metabolites are small molecules made by the metabolism, okay, but they are not absolutely required for the organism to survive. So these secondary metabolites, they are not involved in the growth and survival of the organism. Okay, in other words, you can say the organism can live without them, but these molecules often give extra advantages like protection or attraction. In plant, these metabolites help with the growth and development. So they might not be essential for basic life, but they support the plant in growing stronger or adapting to its environment. For example, some secondary metabolite help plants defend against insects or diseases. So they provide protection as well. Finally, secondary metabolism also supports primary metabolism, which is a set of processes needed for basic survival. So primary metabolism are the processes which are required for the survival of the organism. Okay, clear? Moving on to the next question, drugs and alcohol give short-term high and long-term damages. What will be the correct answer? Okay, so in this table you can see the short-term effects and the long-term effects. And the short-term effects, you can see drugs and alcohols might make someone feel relaxed or lower their inhibitions, a bit like taking the brakes off of the car. So you're removing the inhibition. Okay, so person feels quite free. The person feel relaxed. Sometimes the person feel more energetic, and there is a sense of euphoria as well. Okay, clear. But the person will have slow reflexes because these drugs, they affect the central nervous system. There will be reduced coordination as well and impaired senses and perceptions. Now, what are the long-term effects? Okay, so these drugs, they can seriously harm the body, they can damage the brain, liver, and other organs, and even leading to conditions like alcoholism or fetal alcohol syndrome. Okay, so these drugs, if they are taken for a long time, then they can disrupt the brain development, they can cause liver damage and liver cirrhosis, which is irreversible. Now, it can also cause the brain cell death, organ damage, and ulcers, cardiovascular problems, reduced male fertility, nutritional deficiencies, alcoholism, death, as well as fetal alcohol syndrome. Next question, diseases like dysentery, cholera, typhoid are most common in overcrowded human settlements. Why? So let's have a look at the answer. So you can see here dysentery, cholera, and typhoid, they are most common in crowded settlements because they are infectious disease and spread from person to person. Now, another reason is contaminated water. In overcrowded areas, sanitation systems often cannot keep up. So water can get mixed with the waste from the infected people. Okay, when the other people drink this water, the infection spreads further. So this shows us how clean water and good hygiene are essential for preventing these diseases. The next question, from which plant cannabinoids are obtained, and name any two cannabinoids. Now, which part of the body is affected by consuming these substances? So let's have a look at the answer. See, cannabinoids are obtained from inflorescence of the plant Cannabis sativa. Okay, this is a flowering plant. Now let's talk about some of the examples of cannabinoids. So marijuana, hashish, charas, and ganja. They are all different forms of cannabinoids. Now let's talk about these substances, how these substances affect the body. So cannabinoids interact with special receptors in our body called the cannabinoid receptors, which are mainly found in the brain. Finally, it's important to know that consuming cannabinoids doesn't just affect the brain. The cardiovascular system, which include the heart and the blood vessels, can also be harmed by these substances. Next question, in the metropolitan city of India, many children are suffering from allergy or asthma. What are the main causes of these problems? Give some symptoms for allergic reactions. What will be the correct answer? Okay, so let's have a look at the answer. In big city, the lifestyle can actually lower our immunity and make us more sensitive to the allergens. Another major factor is pollution. The air in cities often contains more dust and pollutants, which increases the chances of children developing allergies. Now, some common symptoms of allergic reactions include sneezing, watery eyes, a running nose, and difficulty in breathing. Okay. Now, next question. What is the basic principle of vaccination? How do vaccines prevent microbial infections? And name the organism from which hepatitis B vaccine is produced. Let's have a look at the answer. Let's talk about the principle of vaccination here. So, vaccination, okay, in what vaccination is done? In vaccination, attenuated pathogen or weakened pathogen or the antigens of that pathogen is injected into the host, and the immune system will generate a response against this antigen or the weakened pathogen, and it will produce, okay, antibodies against that. Clear? And whenever there is a subsequent exposure to the same pathogen, a heightened immune response will be generated. This heightened immune response generates because of the memory cells. Okay, so vaccination is based on the principle of memory of the immune system, means the immune system resembles the antigen, the invader. So in vaccination, a preparation of antigenic proteins or inactivated or weakened pathogen is introduced into the body. Now when these antigens enter the body, our immune system responds by making antibodies and also creating special memory cells called the B cells and the T cells. Now whenever there is an attack by the same pathogen again, the existing memory B cells and T cells recognize the antigen and overwhelm the invaders with massive production of lymphocytes and antibodies. So this is the secondary response. This is highly intensified and it overwhelms the invaders. Okay, clear? So hepatitis B vaccine is actually produced using yeast. Yeah, so scientists use yeast cells to make the proteins needed for the vaccines. Clear? The next question is, what is cancer and how is a cancer cell different from the normal cell? Okay, how do normal cells attain cancerous nature? These are the questions. So let's have a look at the answer. See, an abnormal and uncontrolled division of cell is termed as cancer. So cancer is when cells in the body start dividing in an abnormal and uncontrolled way. Now, genes called cellular oncogenes and proto-oncogenes present in the normal cells, when activated under certain conditions, lead oncogenic transformation of the normal cells leading cancer. Okay, so our cells have special genes called the proto-oncogenes which acts like switches to control cell growth. But if these genes are activated in the wrong way, then they can turn normal cells into cancerous cells. So this process is called oncogenic transformation and it is a key step in development of cancer. Now cancer cell is different from the normal cell in the following ways. Okay, this is a table showing a difference between cancerous cells and the normal cell. So cancer cell they divide without any control, while normal cells only divide when needed. Now cancer cells they ignore signals to stop growing, which is known as loss of contact inhibition. So cancer cells they have lost the ability of contact inhibition, whereas the normal cells they show contact inhibition, means whenever the cell they come in contact with each other they will stop dividing. Also cancer cells can live much longer than the normal cells. Clear? So these are some of the differences between cancerous cells and the normal cells. Next question. A person shows strong, unusual, hypersensitive reactions when exposed to certain substances present in the air. You have to identify the condition. You have to name the cells responsible for such reactions. Now, what precaution should be taken to avoid such reactions? Let's have a look at the answer. If a person is hypersensitive to certain substance present in the air, he may be allergic to it. Now, let's see what happens inside the body. Special cells called mast cells are responsible for these allergic reactions. Now, when they detect the allergens, they release chemicals like histamine and serotonin. To prevent these allergic reactions, the best precaution is to avoid the allergens that trigger them. For example, if someone is allergic to pollen, they should try to stay indoors during high pollen seasons. Next question. What are the lifestyle diseases and how they are caused? You have to name any two such diseases. Let's have a look at the answer. See, lifestyle diseases are linked to things like our food choice, how much we move, our work habits, and even stress. Examples are cancer, alcoholism, heart disease, etc. So these are the lifestyle diseases. So these diseases, they are depending on what you are eating, okay, what is your diet, how much physical activity you are doing, and the level of stress also, because stress can also cause a lot of diseases. It can cause hormonal imbalance as well. Here, moving on to the next question. If there are two pathogenic viruses, one with DNA, other with RNA, which would mutate faster and why? So let's have a look at the answer. The RNA viruses mutate faster than the DNA viruses. So RNA, it is less stable as compared to DNA. So why does RNA mutate faster? Because RNA is copied with less accuracy, and mutations, they happen more often in the RNA. So DNA is more stable as compared to RNA. Basically DNA has deoxyribose instead of the ribose sugar. So it lacks an 2' OH. That's why DNA is more stable. Okay. And then DNA consists of thymine instead of uracil. That's also makes DNA more stable. Okay. Now DNA has special repair mechanism also. Where if by chance a wrong base is incorporated, then the DNA repair mechanism can, you can say, repair it or fix it. So this makes DNA viruses less likely to change quickly. Okay, so the stability and repair system in DNA help keep its genetic information more consistent over time, while RNA viruses keep changing and adapting. Next question. Drug like LSDs, barbiturates, amphetamines, etc. are used as medicines to help patients with mental illness. However, excessive doses and abusive usage are harmful. Enumerate the major adverse effects of such drugs in the human. Let's have a look at the answer. See, here is a list of harmful effects of drugs like LSD and barbiturates. One of the first signs of drug abuse is anxiety, shakiness, nausea, and sweating. People may also lose control over their thoughts and actions. Next, these drugs can cause reckless behavior, vandalism, and even violence. Vandalism means destroying the public property. This means people might act without thinking about the consequences, sometimes harming themselves or others as well. Another effect is the lack of interest in personal hygiene, along with changes in weight and appetite. Now, people may stop caring about their appearance or health as well, and their eating habits can swing wildly, means it can change. widely. So this shows how drug abuse can disrupt even basic self-care routines. Now, withdrawal, isolation, depression, fatigue, and aggressive behavior are also common. People might pull away from friends and family, feel constantly tired, or become easily irritated. Now, social adjustment problems often develop as a result. This means people may struggle to fit in at school, work, or in their community as well. Now, withdrawal symptoms can be severe and even life threatening. When sometimes try to stop using these drugs, their body can react very strongly, sometimes in dangerous ways. Finally, taking excessive doses of these drugs can lead to coma or even death, often due to respiratory failure or heart failure or bleeding in the brain. So this highlights just how serious drug abuse can be. Next question, what are the recombinant DNA vaccines? Give two examples of such vaccines. Discuss their advantages. So these are long answer type questions. So let's have a look at the answer. See, recombinant DNA vaccines. Okay, what are these? So recombinant DNA vaccines are made up of small circular DNA known as plasmid that has very tiny piece of pathogen DNA incorporated in it to produce one or two specific proteins of the pathogen. So what do scientists do? Scientists take a small circular piece of DNA called the plasmid and insert a tiny piece of DNA from the pathogen into it. So this allows the plasmid to carry the instructions for making just one or two specific proteins from the pathogen, not the whole organism. Now this recombinant DNA is placed into bacteria or yeast cells. Okay. Now this you can say this recombinant DNA, okay, it will use the host machinery to make copies of this, you can say, inserted DNA. Clear? So you can see here, this recombinant DNA is introduced into bacteria or yeast cell where it can use cell's machinery to produce polypeptides of the pathogen. So polypeptides of pathogens are being made here, which can be used as an antigen. Okay, these polypeptides of the pathogen called as antigen, these can be used to generate immune response in the host. Okay, so we are only, you can say, targeting a particular region of the pathogen. Yes. Now, these polypeptides of the pathogen are used as vaccine to trigger a range of immune responses. Okay, so this is how vaccines are being prepared now on large scale, because we can now make these antigenic polypeptides in these yeast and bacteria. Now, vaccine produced by using this approach allow large scale production. Example is hepatitis B vaccine is produced from the yeast. Okay, and bird flu DNA vaccines. This is also another kind of vaccine. So these are clear examples of recombinant DNA vaccines. What are the advantages of recombinant DNA vaccines? So recombinant DNA vaccines are advantages over killed or attenuated vaccines since they does not get virulent or mutated again as it is seen in the case of attenuated vaccines. So now we are only introducing a part of the pathogen. We are only introducing the antigenic polypeptide. We are not introducing the weakened pathogen here. Okay, so it is more safer. Another advantage is purity and specificity. Now these vaccines are highly pure and target specific parts of the pathogen, which means they can trigger a strong and focused immune response. Okay, so this was the end of this session. I hope this session was quite knowledgeable for you and your concepts are clear. Now we'll meet you in the next session. Thank you very much for listening.